Thank you so much. So first of all, I want to say that I'm so sorry that I cannot be there in person. I really enjoyed all the talks today, and I would have follow up with, I probably will follow up with lots of questions over email to people. I am just at the last leg of my sabbatical, so I'm on board abroad right now, and I just wasn't able to make it in person, but at least I can join over Zoom. Um, I'm very happy for this talk to be interactive, and feel free to ask me questions. Uh, Even just speak up, and I think I can hear the room, so feel free to stop me, and this can be more of a discussion. So with that, I'm here to tell you today about some of the work we do in my lab related to spatiotemporal patterns that we observe in insect swarms. And we already heard lots of interesting talks today about collective behavior, so I don't think I need to give much of an introduction to the topic. I will just say that in my lab, we focus on communication and kind of keeping in mind the long-range communication we can do as humans with the technologies that we have, asking the same questions about animals, living organisms, and how they solve communication with the resources they have at their environment. So just to show you two very famous examples of that use very different communication channels. This movie here at the top, that's slime mold. I'm sure many of you have seen this before. All of these white blobs are food sources, and the slime mold is essentially exploring and looking for food, recruiting parts of the slime mold with chemical communication in this case, and essentially solving the traveling salesman problem in a fairly decent time. And then another example that we've already seen today, I think a couple of movies of these beautiful murmurations. And in this particular case, they tend to use visual communication to coordinate these really sharp turns to evade predators in this case. So this is the area that my lab is working at, and we're really interested in how organisms, first of all, choose an optimal signal modality, how they integrate the information from all the other individuals in the group, and how do they respond. And then since my background is in physics and computer science, we think about things like detectability, how to increase signal to noise ratio, how to compress information, how to ensure the network is robust to failure of individual nodes, etc. And we work on a really fun, I would say, variety of different organisms. This talk is going to focus mostly on fireflies and honeybees, but we also work on ants, on worms, on plants recently. So if anybody's interested in any of these other systems, I'm happy to talk about them offline. And what I thought I will do today is give a few examples of the problems we work on in the context of fireflies. So I'll talk about things like how do they synchronize with each others, which actually relates nicely, I think, to the last talk we heard about the dolphins. And then also think more about which signals to produce, and all of this will be in the context of visual communication. And then I'll move to talk about bees, where because they're more dense, the communication is going to be more mechanical. And we're going to talk about how they aggregate and maintain mechanical stability, etc. So I'll start with fireflies. And here is a movie of a firefly. It's flashing. It has a lantern in its abdomen. It's called light, bioluminescence. And primarily, it's being used for mating purposes. And the idea is that one firefly would produce a particular flash pattern, and another firefly might respond in a particular flash pattern. And there's many species of fireflies. I think there's over 2,000, and about 70% of them are using these kind of flash patterns to communicate with each other in the context of mating. And here are just a few examples to give you a sense of the patterns that they use. So each line here is a different species. The males of a particular species would have a particular species-specific flash pattern, and then the females would also respond in a female-specific flash pattern. And even though this is a very simple communication signal, it's somewhat discrete, similar to Morse code. When you have many individuals flashing within a swarm and you don't see them when they're not flashing, it's really hard to say which flash came from each individual and then maybe say something about their species based on this dictionary that we know. And one way that fireflies solve this problem is by synchronizing their flashes that immediately gives them an increase in the signal-to-noise ratio. So here's just an example of this behavior. This is a very nice artistic video. The rest of the videos I'm going to show you from my lab is not as beautiful, just a heads up. Oops, sorry. Okay, so the story with synchronization has been studied for a very long time. We know that it's a prevalent behavior in nature, both in living systems, but also non-living systems. And one of these good illustrations of synchronization in non-living systems is the movie that I'm going to show you now. The idea is that each individual, whether it's a firefly or in this case it's a metronome, is doing some periodic behavior. The metronome is going to move right and left with a certain period. The firefly might flash on and off with a certain period. And when you have two of them nearby each other, it's not guaranteed that they're going to synchronize, but if there is some coupling, either social coupling or mechanical coupling, they might become synchronous after a while. So I will try to... I don't think you can hear this movie very well, but I will play it, and if you just focus on the visuals, I think it's just as... is going to make the point anyways. So what we have here is a bunch of metronomes. They are not synchronized when this movie is starting, but eventually they will become synchronized. And that's because there is some mechanical coupling here. And small vibrations from one metronome are going to propagate through this flimsy platform and interact with the rest of the metronomes. And eventually they're going to reach this synchronous state. So that's just a visual illustration. There's, of course, a lot of math models that also explain this behavior in more rigorous ways. One very famous model is the Kuromoto model, where each individual agent in the system has a particular period and also a phase. So if you want to represent their behavior as a function of time, you can plot the phase as a function of time. And then the main equation to couple the individuals in the group is shown here in the first line. The idea is that the change in the phase of one individual is, first of all, related to its own frequency. That's this omega element here. But then also it's coupled to the phases of the other individuals. And that's this sum over a sine element here. And you can have a coupling coefficient. It's written as K here. It can be lots of different things. Usually it's also a function of the distance between individuals. And that's because if two individuals are further away from each other, if we think about fireflies, especially when their light is so dim, then they might be less coupled as they become further away from each other. So when I opened up my lab, I was really excited about this firefly problem, and I realized that there's not a whole lot of quantitative data to try to relate the firefly behavior to these very nice mathematical models for fairly good reasons. First of all, fireflies exist as flashing adults for about 2 weeks out of the entire year. So you really have to be at the right time to find them and to find these synchronous swarms. And also, the technology wasn't quite there until recent years. Nowadays, we can even go with our smartphones and record the flashes of fireflies pretty easily. So that was the idea, really just to try to fill in this experimental gap. And myself and Julie Hayes, who was a student in my lab, and Raphael Sarfati, who was a postdoc in my lab, went to the Smoky Mountains in Tennessee, in the US. And we worked with a species of fireflies that flash in bursts. So they flash a couple of times, and then they stop as a collective for a while, and they flash again a couple of times. And this is an example of a recording. So we're positioning one of the cameras. This is the synchronous behaviors. It's not perfect, and I think that actually makes it more interesting. But I think you can clearly see that there is some interaction between the individuals, and there's also some propagation, spatial propagation. And we really wanted to capture that. So we positioned our cameras in a stereo configuration. And from there, we can actually do the 3D reconstruction. So we can get the particular time and also space, position in space where we see flashes. And here is an example of such a reconstruction. We can get a lot of flashes. This is a recording that was taking place over, I think, about 15 minutes out of the entire night. And we can start classify a little bit more rigorously the transition between non-synchronous to synchronous behavior. And I'm going to start here with the plot actually at the bottom and then explain the plot at the top. So at the plot at the bottom, we see the y-axis is just the number of flashes that we record as a function of time. And early in the night, we see the plot on the left. So there's not a lot of coordination between the individuals. And then later in the night, we see the plot on the right, where we start seeing these burst structures, flash patterns also on the level of the group. So if we characterize things like the variance of the signal over a time window that is rolling through the night, and plot the variance as a function of the average, this is what we're seeing here at the top plot. And when we have low flash density, then we're seeing something that has a characteristic of Poisson distribution. So it's a random process. And then at high density of flashes, we're seeing something that is more related to the clustering of the flashes when they all occur at the same time. And we can see this reflected in basically the exponent of these two properties. So there's a different scaling law, and somewhere here in the middle, that's where the transition happens. Okay, so that's a temporal thing. In terms of space, we can capture things like the propagation of the onset of the burst across the field of view that we record. So what you see here is an overlay of all the flashes we saw from one camera view. The early flashes within a burst are shown in blue, and then later on are shown in yellow and in red. And you can see that there is a propagation from the top left to the bottom right of this figure. We can of course measure the speed of the wave, and we can actually also measure how the individuals are moving while we see this wave, and we see that it's completely uncorrelated. What we see is that individuals move randomly in terms of the direction, uncorrelated to the direction that the wave is propagating. And that tells us that we see this wave not because individuals are moving, but because there's some kind of interaction between individuals that triggers that wave. And then another thing that we can do to characterize the spatiotemporal correlations is to plot histograms of, like I'm showing you here at the top right. So this is a histogram of all the pairs of flashes as a function of how far they are from each other in terms of space, in terms of just spatial coordinates. So that's the y-axis, and also as a function of time. And there's a couple of different interesting patterns here. I think these red blobs that you see here at the bottom are actually single fireflies that we're seeing being correlated with their own pattern. But we can also see the correlation between different fireflies, and that's here at the top. You can see how it decays and so on. Personally, I was really excited about this because I thought that now that we have the spatial aspects of this, in a sense, representation or some kind of proxy to maybe the coupling in terms of space, we can now go back to the mathematical models and plug that in. But the situation was a little bit more complicated. And we found that when we did experiments with individual fireflies. So if you remember from the mathematical models, one of the core assumptions is that individual fireflies act as some kind of oscillators. So they have some periodic behavior. And when we track individual fireflies in more controlled environments, here we have basically just big tents that we bring in some fireflies into, we found that that's not exactly the case. So we have an experiment here comparing the behavior of a single firefly and a larger group, 15 fireflies. And this is again the time series, the number of flashes as a function of time. And every time you see a peak here, that's actually a burst. So for the 15 fireflies, when we zoom into one of these peaks, we see a burst structure. Also for one firefly, if we zoom in here, we see smaller bursts because it's just individuals, but still bursts. The interesting thing is the time between bursts, which is very consistent for 15 fireflies. You can see it by eye, basically, that it's pretty punctual. It's actually every 10 seconds or so. But for one firefly, this distribution is much, much wider, which got us to question the assumption that maybe an individual firefly acts as an oscillator, and that really complicated things. So we're in the process of trying to develop new models. I don't have it here in my presentation, but I'm happy to talk to you about how we're approaching this. And yeah, if we have time later, I can maybe even show you a few results. Okay, so I think I might skip this just for the sake of time. And get to another question that is slightly more broad. So I just showed you one species of fireflies and how they deal with other individuals in their environment. They synchronize, they immediately increase their signal to noise ratio. But I also showed you that there's many species of fireflies, and actually many of them sometimes coexist. So they essentially need to deal with also having coexisting flash patterns and trying to distinguish between them. And that's a really interesting problem that we are trying to better understand also in the lab, not just for synchronous species, but more generally for firefly communication. And to try to tackle this problem, we developed a very simple model of how these flash patterns may have evolved. And I really feel like I cannot stress enough that this is a very, very simple model. It's totally a toy model, and I think we're still pretty far from relating this to real data. But hopefully I'll convince you that even with this really simple toy model, we're getting some interesting flash patterns that seems to be biologically relevant, and how we're planning to take it from there. So to try to think about the problem of designing distinguishable signals, we define a cost function. And that's something that Chantal, who used to be a postdoc in my lab, worked on until recently. We have two elements in the cost function. We have the first element, which is a measure of the pattern similarity. And there's lots of different ways to define that. We chose to consider the longest common sequence between the flash pattern of one species and a second species. And because we're not really dealing with strictly synchronous species, and also there's no guarantee that all species would start flashing at the same time, we're also making sure that we're shifting as a function of time all of these flash patterns relative to each other, relative to all the possible shifts in the starting time. Okay, so that's the first element. And the second element is a measure of energetic cost. That's one way to look at it. You can also think about this as maybe predation risk, in the sense that whenever the firefly is flashing, then it's either costing energy or it's more exposed to potential predators. And here the definition is also very simple. We're really just considering the number of bits that are on. So in the top example, it's 12, and in the bottom example, it's 8. Okay, so that's the cost function. And now we're just going to use this cost function to run a evolutionary simulation where we start with a particular set of species and some random flash patterns, and then we mutate them and accept the new flash patterns if they reduce the cost function. And we basically repeat this as a function of time until we get to some convergence of the flash patterns. Okay, so here is an example of such a simulation. Here we have 6 species, each one is in a separate line, and I'll just let this play. In the early stages of the simulation, we're getting fairly random patterns until it kind of snaps, and then we're starting to get more periodic patterns, and we even see bursts. So seems quite similar to what the fireflies produce in terms of flashes in the literature that we know. Even though we didn't explicitly impose the sequences to be periodic, so it's kind of an emergent outcome of the rules that we defined. So now, just as a small exercise, Chantal basically took a phylogenetic tree of some of these species of fireflies. We know what is their flash patterns from the known literature. So that's what you see here at the top right. And then the idea is to maybe use this simple simulation to try to infer some of the driving forces, evolutionary driving forces of the design of these flash patterns. So the idea is to run a lot of forward simulation with a parameter sweep over the 2 parameters, or actually 1 parameter that we have, which is the ratio between these 2 parts of the cost function, and then compare the patterns that we get from the simulations back to what we know from the experiment, and try to find the minimum there. Okay, so that's the idea, but we're actually missing some data, I would say, missing quite a lot of data. So overall, generally speaking, the data about different species of fireflies and where they are is fairly sparse these days. And we're trying to get more data. It turns out that the methods that we use to record fireflies for our own experiment in the lab in the field, we kind of made it easy to use for us, but it's also seems to be fairly easy to use for potential community or citizen scientists, where instead of the more sensitive Sony cameras that I showed you before, we're now using GoPro cameras. And the idea is to simply put the GoPro cameras somewhere where there's fireflies. No need to deal with any calibration. We can do that in a post-processing step by using the firefly flashes as a landmark. And then we are trying to create this bigger database where we'll have more information about what different species are doing. And we also have some new work where we train the machine learning model to basically get as an input some random flash pattern, and then it can tell us which species it's most likely that was being reported. So here are just a few examples from this citizen science project where people helped us record. You can actually see that there's quite a lot of variability within the species, and that's an interesting aspect that I'm happy to talk later about. And yeah, if you happen to have known locations of fireflies, maybe in your backyard or somewhere near where you live, and you're willing to help us, please let me know, and we'll be happy to sign you up for a kit. Okay, that's all I wanted to say about fireflies. And yeah, I think I'll move to tell you a little bit about the work that we do with honeybees. So far, the communication between individuals was very visual. And with honeybees, they have they're going to be more dense, so we're going to encounter some chemical communication and some mechanical communication. They have a very different hierarchy in comparison to the fireflies. Honeybees have colonies that are composed mostly of female workers. There's some small amount of drone males and one queen, usually. And she's very important. She's the only one that can lay fertilized eggs. So the rest of the bees tend to aggregate around her. And presumably that's based on pheromones. So some of the bees can recognize the queen based on her pheromones, but pheromones is something that decays very rapidly in time and space. So bees that are really far away from the queen wouldn't have a good chance to pick up these pheromones. So the bees solve this problem in a really interesting way. They basically, each bee that, I'm going to show some examples of that in a second, each bee is going to serve as an amplifier to the signals to the pheromones of the queen. And it works like this. So honeybees have this little gland here at the tip of their abdomen that is shown in with the red arrow. They can change their body posture to open it up, and then they fan their wings, basically draw air backwards across this pheromone gland, and move these pheromones backwards towards the less informed bees. And this is just a cartoon of how it works, and I'll show you some real data in the next slide. So if we have a target and a bee, one of these green bees is noticing it, she's going to start producing these pheromones backwards. It's, by the way, called scenting because they produce scent. So this bee is going to scent backwards, and then another bee is going to sense the pheromones of this first bee and start scenting also, and another bee and another bee. And so they create this really interesting network of communication that transfer the information about where the queen is over many, many body length of the individual bees. Okay, so we're trying to better understand how the bees decide when to transmit this information to the rest of the bees and how do they orient themselves. So we ran some experiments in the lab, and this is a two-dimensional system. The queen is here in a cage. So she can't really move much, but the rest of the bees can still smell her and use her pheromones as a signal to start this process. And so the queen is going to be up here at the top right. The rest of the worker bees are going to be here at the bottom left. And every time you see one of these blue arrows, that is a scenting event. And it also shows you in which direction the scenting is occurring. So I'll play this analyzed experiment. You can see that there's lots of arrows, which means that there's lots of scenting. And if I scroll a little bit faster through this movie, after something like, I think, 15 minutes on average, they do find their way to the queen and aggregate around her. Okay, so we wanted to, yes, this movie. Okay, so first of all, we wanted to make sure that these scenting events are meaningful in the sense that they show the way to the less informed bees how to get to the queen. And the way that this was done was to take a snapshot from the experiment. We take all of the scenting events and we treat them as little gradients that we then integrate into a higher dimensional surface. And the hypothesis was that if indeed these scenting events are meaningful, they would integrate into a surface that is high, where right where the location where the queen is. So here is an example from one experiment, and you can see indeed that the surface is higher where the queen was located. If we do a control and we take all the scenting events, but we randomize their direction, we get a completely flat surface. So that tells us that the scenting events are, in a way, creating these sort of a global map that shows the way to the queen. Another interesting clue from the experiment about the decision making was that the bees that are scenting tend to position themselves at a particular distance from each other, roughly 6 centimeters, besides a couple of outliers that we have at very, very sparse systems. So if we're talking about some kind of diffusion from a source, and the next bee that is scenting is always positioned at a particular distance from that source, that implies that maybe there is a concentration threshold that triggers that behavior, because at a particular distance from the source, we're expecting to have a certain concentration of the pheromones. So we take all of this together now and try to create a simple agent-based model of this behavior to try to understand how some of the behavioral parameters, like the threshold that triggers the scenting behavior and also the directional bias of the signal that is caused by the fanning of the wings of the bees, how these two parameters affect the aggregation process. So the model is, like in the experiment, it's a two-dimensional arena. Bees can kind of walk randomly in this arena until they bump into a cell that has a high enough concentration of pheromones. And then they have two options. First of all, they can go up the pheromone concentration, Or they can also stop and produce their own pheromones to inform the rest of the bees. So the scenting behavior is looking like this. We're basically solving the classical diffusion equation with some directional bias, and then we have a knob that tells us how directed the signal is. Okay, so we can now run a nice parameter sweep, and this is what you see here in this phase diagram. The y-axis is the threshold of pheromone concentration that triggers the behavior of scenting, and then the x-axis is a measure of the directional bias. So 0 means that it's a completely isotropic signal, and high values mean that it's there's a high directional bias, so it's very directed. And we're seeing a couple of different phases here. First of all, if the critical threshold pheromone concentration is too high, then of course nothing happens. Nobody is scenting, nobody's aggregating around the bees, around the queens. And if it's too low, then they tend to get stuck in small clusters in a state that seems to be some kind of local equilibrium. We also have some diffusion limited aggregation where the bees kind of move randomly until they bump into the surface of the cluster around the queen, and that scales really badly with system size. And then we have this more kind of like a sweet spot where the bees are able to transfer the information about where the queen is across the system size and also aggregate into one coherent cluster around her. And I always like to stress out here that if you're familiar with aggregation processes, then these phases are not really surprising. You can see it in many different systems. But I think what's really interesting is that this more optimal phase that allows the bees to efficiently aggregate around the queen opens up with directional bias. And that indicates that the directional bias is indeed a positive thing when you are trying to aggregate around the source. And that's really the bottom line here. All right. So what I just showed you was a very nice, clean, easy, air quotes, experiment where the bees were trying to aggregate in a two-dimensional environment without any obstacles. But in reality, bees can also, and they do so very often, they aggregate in 3 dimensions. And when you start adding the 3 dimensions and gravity, the problem becomes much more complicated, of course. So this is the kind of structures that I'm referring to. This is really just a structure that is made of bees. The bees are not flying, and there's nothing really here besides bees and a tree branch. So the bees are really clinging on to each other. This is a structure that occurs as part of the reproductive cycle of European honeybees. And they stay like this for a few days, usually until they find a more permanent place for their nest. And they're completely exposed to the elements. They don't really have a nest structure to protect themselves from changes in temperature, for example. So when the ambient temperature changes, they have to adapt to stay warm. And they do that by changing their density, for example. So here you see at the top 2 pictures here, it's the same swarm, but at different temperatures. So when it gets colder, then they become more dense to maintain their temperature. When it rains, they tend to orient their body according to the direction of gravity. And it's called the shingle-like configuration because it makes the swarm less susceptible to water. And at really high temperatures, it's been speculated that they create these really interesting channels that helps them exchange air with the environment. So it's a very dynamic structure. It responds to the environment when we change the temperature, for example, or with humidity, rain, and so on. And it needs to do all of that while maintaining mechanical stability. So this, dealing with gravity is dealing basically with a constant dynamical force in one direction, and different shapes of the branches. Because of that, may result in different shapes of the swarm. You can see here a few examples. But it also needs to deal with more dynamic forces. And here is a movie of a swarm on a windy day. If it's going to get shaken too much, it might break, and that can be pretty bad for the bees in terms, it basically kills the thermoregulation purpose of this structure. And it doesn't happen very often. And basically, we wanted to understand how come the swarm doesn't break. So it's a saga that started probably almost 10 years ago when I was a postdoc with Mahadevan at Harvard University, and he had a really good idea to try to apply some mechanical perturbation to these swarms and see how they respond. But essentially, it really means that we were shaking honeybee swarms and just seeing what happened. So this is an example of this experiment. And we saw that once we shake them for a while, they actually change their bonds. They move around within the swarm collectively, and they change the shape of the swarm to become more mechanically stable. So in this particular case, they start with a certain cone-like structure, and then they spread out. So they reduce the height of the cone and they increase the base area. And we also had an agent-based model to explain this behavior. We explained it by the bees following local signals that are related to how the bonds they make to their neighboring bees stretch over time, and actually go towards a direction where bonds are stretching more to stabilize the swarm. And then all of that information was known just from looking at the shape of the swarm from the outside. We could even track some individual bees, and we can see them kind of crawling upwards. But really, the bulk of the overwhelming majority of the bees is inside the structure, and we were not able to see what's going on inside until recently. So that's a collaboration with Kaushik Yararam from CU Boulder also, and we have access to an X-ray machine where we can put these swarms inside an X-ray machine and then start to peek inside and see what's going on, also in the bulk structure by doing a 3D reconstruction. What we found, I mean, we started looking at something like the scaling law of strength versus body weight. We know there's some very nice work published on these scaling laws, and these are actually taken from this is a paper from the 50s. It's was looking for a nice, convenient data set to try to relate body weight to strength. And so they worked with information about weightlifters. And they saw that if you plot the maximal strength that one of the participants could lift versus their body weight, then you get a very clear scaling law, this behavior. So we were trying to find something, a similar signature of that in the, okay, good, thank you. In the honeybee swarm. But because we're now dealing with a group of bees, we had to define the problem a little bit differently. So instead of an individual bee, now we're thinking about a slice through the swarm and how much weight it can support underneath. And that's something that we can measure because we have the X-ray reconstruction. So the plot here at the bottom is showing exactly that. So on the y-axis, we have the weight that each layer can support versus the mass of the bees in this layer. And we're also getting a scaling law with a different exponent that I'm not going to go into the details of that, but we have some continuum mechanics theory to explain this different exponent. And all of this data that you're seeing here with the different colors is coming from swarms of different sizes. So they all seem to be falling on this somewhat universal curve. Okay, so just as a future direction, we can also get some time lapse of these structures. And you might be able to see that there's areas in the swarm that are more dense, areas that are less dense, and they kind of fluctuate as a function of time. We're kind of wondering if this is maybe a signature to the model that was hypothesized in the 80s, and that's something that we're currently working on in the lab. Okay, so I had another problem that I wanted to talk about, but I think that maybe given the time, I will just say that my lab is also working on how bees, as a collective, manipulate inanimate materials like wax and how they build their beautiful honeycombs. And we are basically tricking them into, we're giving them an impossible puzzle by 3D print very annoying honeycomb frames from them. So of course, when they build their own frames, they tend to build very pristine, homogeneous, to some extent, honeycomb structure. We're giving them problems where they're not able to do that, to see how they deal with some frustration. And so we create two patterns, two arrays, but we tilt them a little bit, or we shift one up, one down, and then we see how they deal with this frustration. I'm not going to go into details here because I'm running out of time, but if anybody's interested in that, I'm happy to chat later on. And I think I'll just put on the most important slide. Everything I showed you was amazing effort from an amazing team of grad students, postdocs, undergrads, beekeepers, high schoolers, and collaborators. So I want to acknowledge all of them and thank you for your attention, and I'm happy to take questions.